So we can start.
Okay, thank you. Welcome you all again. And today we're going to talk about again the synthetic biology, and today is this lecture 2. So I'll go a little recap about from the last lecture, and where we actually talked about a few of the potential applications of the synthetic biology, along with how the idea of the synthetic biology is coming, that design-based biological systems, how it is coming from various aspects of evolution of the biological sciences and technology. And today we'll talk about some more applications, projected applications, some of the fields where synthetic biology can or probably can change a few of the things which cannot be possible probably through the conventional biotechnology and the genetic engineering without that design. And then we'll see a simple case study that how we are going to develop a simple synthetic genetic circuits. What would be its chemistry, what would be its physics, what would be its engineering, and what could be its mathematical modeling. So we'll try to keep everything in simple for everyone, and then we can always go in detail in the next class or so. So again, I'd just like to tell you, in the last class, I just showed this picture to show that what is the complexity of the biological system. And I talked about that. If you think about this biomolecular network of the bacteria, which is similar to the complex as the computers connected with the internet in 2009. And so, and then there is a lot of the interactions among those particular nodes. These nodes could be the genes, could be metabolites, and could be proteins, and so on. And so there's a lot of feedbacks in those systems. There is all interactions, at least most of the interactions are nonlinear, and there is a lot of noise. So the biology is working through a very complex soup of the chemistry, and there is a very interesting connections among them. And that's actually give us to think that how we can study the biological systems or a biological cell from a systems point of view. And this is the starting point of the philosophy of the synthetic biology, where last week again we talked that we do not know how from a protein to genes, from biochemical reactions, pathways, and how these are connected together. The rules at different scales, we are not very sure because it is so complex. But we have seen that, at least we've seen that if you take some of the principles from the electrical or electronics engineering and try to apply in the biological networks, try to explain their properties, you can actually explain them up to an extent that you can do that. And so that is the starting point of the synthetic biology. So whole idea that you want to develop new systems in new biological systems or biological pathways or something, following those rules means you take the protein and genes, and then you develop a biological gates in terms of biological reactions. Okay, so when your biological reactions in the terms of the gates are there, we do know from the electronics engineering how these gates can be assembled and actually develop a computational function. Similarly, you follow the same rules. If I biochemical reactions, if I develop an AND gate, one NOT gate, one OR gate, and I know that these three gates, if you in electronics, if you connect these three gates in some way, that so we can develop a particular function. Here, similarly, we just connect those biological gates in a similar way as electronics, and you develop a different kind of the biological computational function, and that is a kind of the pathway, you can say. And so if we can go a more complex and complex thing, then we can actually develop lots of more complex function. And so these functions are new, but if you connect a lot of new functions, we can actually develop a lot of more complicated functions. So that is the whole goal of the synthetic biology. But I try to mention that one thing, it doesn't mean that you can do everything by this kind of the electronics engineering. We take that electronics engineering principle into the biology, because that's a knowledge base the human know very well. Okay, and we can apply those things in the biology for this moment. But biology, as said, it is much more complex. So until we actually understand a specific rule of the biological design, the way the other design principle actually developed in mechanical engineering or electronics engineering or electrical engineering, until then we try to use those kind of the principles. But biology is completely complex, and their biological actual design rule in the biology may be different. We don't know yet, but we are searching for those. Okay, and we also, in the recap, I showed that we actually developed a lot of different, all kinds of possible biological, all the basic logic gates, and nor, so, or XOR and everything, XOR, XNOR and everything. Okay, we actually developed in last 20 years or so. We also showed that if you develop those kind of the gate, you connect and you can develop a lot of different kind of applications. One, toy applications are of those kind of device principle, where you develop a circuit with the mRNA. And if this classifier is called as classifier circuit, if this classifier circuit actually sends the microRNA signature of a HeLa cell, then it actually is turned on and start producing a peptide, a protein, which is going to make this the whole cell towards apoptosis. If it is not a HeLa cell, so that the microRNA signature won't match with the HeLa cell, then this won't turn off and the cell will do as it is doing. Okay, so this is an application we showed. And we also showed this kind of the computational applications for much more complex applications, the way you can actually add numbers using biological cells, basic genetic regulatory network, following the same kind of the principle what you can see in your computer arithmetic logic unit, which has have some adder. So you develop that you take the same principle, the way those gates are connected, and you take this kind of things in the biology, connect those gates, and you can make an error in mammalian cell, which is published like a couple of years back or so. So this is some applications state of art at this moment. So we also saw some of the projected application of the synthetic biology. So this kind of the computation, how we are going to use those kind of biological computation. One area is definitely the program therapeutics, where you can actually program your gut bacteria or a cell in such a way it can sense a states, physiological or disease states, and then it's turned on, and then it do something to actually mitigate that situation. We also talked about just a little bit about synthetic metabolic engineering, where the whole idea is like you take a pathway from other organism. That pathway may give a very interesting chemicals for your use, and that whole pathway, or a part of the pathway, you put directly in the yeast or the bacteria, which you can actually cultivate in a reactor in a controlled environment in such a way. So, and you produce those chemicals which previously you need to extract from the plants or some other animals or something. Now you probably can do that in bacteria or the yeast, and it has an enormous potential because you can develop any chemicals, any drugs, and fuels. And thirdly, we also talked about that it is a little more towards future, that the way we are designed. So somehow from a single cell, we are actually developing our organs during our development. And so that information, that computational information is within our genome, but we do not know how it is happening. So synthetic biology want to track that. I mean, many physicists and many chemists actually track that. As I said, the Alan Turing, the reaction diffusion system. And if you now you can actually hack that, okay, instead of tracking, now, and you try to develop some kind of the pattern, the way the pattern formation is important for developing those organs in vitro. And now we can, those kind of patterns we can actually engineer in a very design-based way. It is mathematically predictable, and you can actually develop not only in the mammalian cell, but in the bacteria. So this means that those principles, what we know about at this moment, what is the physical or engineering principle developing organ, we can actually mimic a little bit in the biological systems, and probably within next, I don't know, 10, 20 years, we'll see some good results on that. So now I'll go that the other applications, projected applications, before going to the case study. So when you're talking about this heart, when you're talking about the heart, it doesn't mean this is a heart. So this way, we can actually engineer the developmental biology, okay, from a very engineering perspective. But when you have that kind of the knowledge that you can actually engineer developmental biology from an engineering point of view or a biological engineering point of view, then you can think about, it's not just the animal, but you can think about the plant. So here is just a toy project. It's the idea is called MIT Vision project, and the idea is to grow your house. What does that mean? So, so you know that when a plant grows in its own way. If it's a bamboo tree, it goes in its way. If it's just a bush, some bush tree, it'll go like a bush. If it is a pine tree, it goes straight or something like that. Now, if we really know the developmental biology, and if we can engineer its development in such a way, so we'll tell that plant to actually grow in our way, in human designed way. If it is possible, then you can actually develop a house, something like, it's not a development, it's a the grow your house. So you engineer this developmental biology of this plant in such a way, one or two or three or several plants will actually grow and give a shape like a house. It's not necessarily it has to be a house, but it could be a bridge, it could be something. So that's the level of the, but we can think about that from now, though we haven't made any progress in that level. But plant synthetic biology is something which is coming up in last few years, and people are trying to develop a lot of very interesting things. Another area very recently people entered, I mean, synthetic biology entered, is called living materials. So, so the whole idea that is the modern city, what we use lots of different kind of the materials from the macro to the nano level. And the whole idea that those kind of materials can be produced by the biological cells or organism in a very programmed way. Okay, and so there are two different level of the scale. One is the city scale, is a very big scale, and another is the exactly in the nano level. In the both cases, synthetic biology can give some applications. I'll just go one of them. It is the nano level engineering. So basically here, people who developed a simple engineered in E. coli. This E. coli is going to produce a peptide, and it comes through its wall, and it's protrude like this chain. Now you can control that peptide's composition, because there is a different kind of unit there. Let's say it's the ABC. Now you should control when it is coming out. It is coming as AAA or ABC or AABB or CC, something like that. Because you give some chemicals and you have some kind of the logic gate through which you can actually generate this kind of peptides. And now you use this peptide, I mean, you choose this peptide in such a way. So there is some gold nanoparticles, like, you know, people who are working on this nanoscience and engineering, their problem is that when you develop some of the nanomaterials, how you actually pattern them in a way. If you want to make a gold nanoware, how these gold nanoparticles you actually keep one by one, and to make almost a single file of gold nanoparticles. So here, if these peptides can bind with these gold nanoparticles, so in this bacteria, if you have those kind of peptides, which is coming like a very programmed way, depending on the its composition, and there, through this composition, you actually control the interaction of this gold nanoparticle and that peptide. So in that way, you can make a gold nanoware and different kind of the patterns that how much depend, I mean, just changing the compositions of these peptides, which is controlled by a logic gate. So in that way, in the nano level, you can develop some the nanoware using a living bacteria, and this is sometimes called living materials. Okay, so this is in the nano level. I would like to show exactly in the macro level, and that is, it's come like just 2 years back, not even 2 years. This is published in 2019, where you want to develop a concrete, okay, and you mix this concrete with some of the bacteria which produce calcium carbonate. So when this produce this calcium carbonate, what happens? This it's called the cementation, the biocementation. So you give some materials like sand, okay, and this bacteria mix it, okay, put in a mold and just keep outside for I don't know some time, and then this bacteria is going to calcium carbonate and actually stitching those sand particles through this calcium carbonate, okay, and this way you can make the concrete that way. So that is as published like 1.5 years back. Um, this one. So this way you can actually make the concrete. But people thinking, I mean, the group who actually developed, they're thinking it's much more advanced. So it is not came yet, but the way people can think about, now you can develop a bridge or a concrete which is self-filling. Means you have some engineered bacteria with that within that particular bridge of the concrete, and if there is a crack, and your bacteria will actually do this biocementation in the crack, and then they heal that crack. And what is going to get is a self-filling kind of the concrete, or self-filling kind of a bridge or something like that. So now in terms of the materials, I just showed 2 examples from nanoscale. You can go to the exactly in the macro scale in the city level. So that's another important area or exciting area for synthetic biology application. And another area which also people think, and that is that what is the potential of the synthetic biology in space. So if you see, this is a poster made by NASA, and their idea that today, so one of the space travel goal, or the human space travel goal, is to go to the Mars, right? But going Mars is not exactly the same as going to the moon. Okay, the first thing that if you see, there's the space travel, or human space travel, in the last 50 years or so, we have seen that the whole idea at the very beginning to show that humankind that we can do something. So we can do something means we can go to the moon and bring the astronauts back. And that's in the huge amount of steps human society or the human science or technology have been taken. Now the goal is not to show that you can go to the Mars and you can come back. You can do that, right? We have the technology, except one thing. So one important part, now we want to develop a research based on the Mars. Because if you want to go to the Mars, it is not a few days thing. It will take years, okay? It may take 3 months to go there with the first test rocket, and if everything goes right, okay? And then you stay there and stay there for 1 day and coming back. It is not going to happen. So whole idea, I want to develop a research base there, okay, where the later astronauts can go and do some work. There's a lot of instruments which will study the Martian atmosphere and something. So if you have seen the movie Martian, probably you have actually seen those kind of the things. So the whole idea, you go and develop a research base. Now the problem, as I said, so going there, coming back, and staying there for 6 months or so, it will take a lot of resources, right? You're starting from your medicine, starting from your food, everything. Now it is almost impossible at this moment to take all the resources to go to the Mars, make a research base, and come back. Okay, so the whole idea of the engineering idea, that whatever the resources you have available on the Mars, you have to use the resources and make your research base. So to do that, it is called in situ resource utilization. And here people believe that the synthetic biology will be probably the key for this kind of the space travel. Because instead of taking a lot of resources with you, you are just taking some vials of engineered organism, okay? Maybe some plants too. And those vials have this engineered in such a way, they will use the Martian atmosphere, whatever the carbon dioxide it has, whatever kind of the vacuum it has, what kind of the small water or something it has, what kind of materials it has on the Mars land. And using that, you start from making the roads using this kind of the biocementation. You develop, say, a lot of regolith biomining. You develop, say, food optimized process. You the radiation resistance materials and everything by those engineered organisms. So that's why sometimes people believe that the key to this kind of the space travel, or the long duration human space travel, synthetic biology is the only key. only key at this moment, at least. So that is one area. And now here you can see that inspired by the synthetic biology, now NASA start thinking about a self-replicating and self-healing building materials. Okay, and it takes the fungus, and this fungus it gives this material, and this is a project. This is a real scientific project in the Ames Research Center in the NASA, and they're trying to develop this kind of the materials. So it is self-healing and it's a self-replicating because you are using the fungus. Okay, and we'll see probably in a few years how this project goes. Another area is not just going to the Mars and those things, but in the space, in the deep space, you basically need a lot of resources, and those resources you can use the sunlight because sun is abundant, and then you need to use the cyanobacteria or different kind of the algae to develop that. You need the synthetic metabolic engineering for that. So there's the two reactor plan. So here it's what it looks like. This is a telescope, but it's a reactor. You can see three different reactors. They have the algae. This is again, it's a toy reactor. It is not placed in the space, but this is just a plan. So here, whatever you are actually trying to develop, from the drugs to the chemicals and everything, you actually develop in this reactor using engineered or synthetically metabolically engineered bacteria. Similar thing people think about as a wall for the space seats, because they want something called a water wall technology. So here, whole idea that whatever is on the wall, instead it will have some you put the algae on the wall so that it takes the sunlight, okay, and then take all the waste materials from the space sea, from the human waste material, okay, and then actually decompose it, and then it produce a lot of chemicals using all those things, and then want to put in the wall because this algae will grow from the sunlight. So this is if you have this synthetic biology, and then you want to develop this kind of the reactor. So this has to go hand by hand. One is this kind of the space bioprocess engineering as well as the synthetic biology together, and probably one day we are going to achieve those kind of things. And again, this is the real project. This is not a toy project or something. So in 2015, Adam Markin from the Caltech and its co-worker, they actually proposed that what would be the grand challenges in the space synthetic biology. So they talked about, and they find there's probably the 6 areas where the synthetic biology can actually give a lot of impact in terms of the space engineering, space bioengineering. One is the resource utilization. That's I already talked about, the in situ resource utilization. You go in the Mars, you do the mining and so on, and using all these kind of using this engineered organism. You can do a lot of manufacturing from the materials to the drugs and so on, because through this engineered microbes or other organism, you develop life support systems for in your spaceship, which have a kind of you take all waste materials, produce oxygen, produce raw materials for things, and so all this kind of thing. You develop the space medicine. That is very important, because if you develop those kind of medicines in the bacteria or something, and then it's a program therapeutics, that is most important. You develop a lot of communication, biological communication system in the space, which is called space cybernetics. Means what is the condition of the radiation, how that radiation is going to harmful for the human, and in the real time, if you can sense that, and you can actually monitor and give some idea about that, that is space cybernetics. And definitely one important goal is that terraforming. So terraforming means you change the climate of, for example, on the Mars, similar to the Earth. So the one day people can actually migrate from Earth to the Mars for living, and that is it's a very long-term goal, but we can think terraforming from others' perspective. At least I do believe, because now the condition is you just not need to just terraform Mars. Sometimes you can think terraforming Earth, because the way the climate is changing on the Earth, the carbon dioxide is increasing and everything. Now probably you can think about some, it's not a full terraforming because Earth is just terraformed, but something of that scale. So if you can actually go some research on this terraforming, then you can find those some of the solutions what people thinking about the terraforming can probably be applicable for our art also. So that's an important aspect of the space synthetic biology. Yeah. So another area, and that is again, it's coming to this biocomputation, because there's one area where you think that a biological cell as a computer would work. So now you probably know the robotics. Everybody's talking about the robotics, right? So there's a lot of robots that are available now. And now one area that how you actually reduce the size of the robot. So this is a small robot. It's called a Spot Mini. It's a dog robot. It's for office. You can take the files and everything. This robot can go to at every table and give you the files, take the files, and go to the such kind of things. Okay. And now people think of the micro robot. So you can bring in the micron level. Unfortunately, no robot can come in the micron level. Okay, a micro robot actually have this kind of the dimension. So this is a penny. So you can imagine this is some centimeters, this is several centimeters in that. But why you cannot bring this robot in the micron scale? Because you need a lot of circuits, you need a lot of decision making circuits, sensor, the actuators, and definitely the energy source, that's the battery. And you cannot bring all those things in just a micron level, so that even the micro robot you cannot bring there. So now think about a cell, a bacteria or a human cell. They're really on the micron level. A bacteria is 2 to 5 micrometer, like E. coli if you think. Now, you do not need a battery for that. You give some food to the environment, they will take that materials and make their own food. And what you need to develop, you need to develop a lot of computation inside the bacteria so that it can sense many things. Okay, and then a lot of decision making circuit, it can take decisions, and using whatever its capacity, you can actually develop those kind of. So that if you bring, so where you cannot bring your computer at that micron level, but you can make a bacteria or a cell as a computer. So this will give, sometimes it is called a cellular supremacy at certain area where you cannot bring, you need a computation, lot of computation, lot of robotics, but you cannot bring a computer or robot in that size range. So that is an important applications people are thinking about. It can have applications in therapeutics from anywhere. We maybe sometimes we like to discuss on that. So if we can all do this, if this is this kind of the applications we can do, so how it is going to change our whole world, the way we know that in terms of the economy, they call this bioeconomy. So this is a white paper from European Union, and you can see the way they are trying to tell you that everything what you today you call as the chemical economy or the material economy, you can change to the bioeconomy. So it could be starting from the food, it could be starting from your fuels, it is a climate change, it is a non-material like materials and everything, and each and every part you can actually change through this kind of the synthetic biology. So it will probably within next 50 years or so, we have already started seeing the transition from the chemical economy to the bioeconomy, and that bioeconomy maybe within the next 50 years or so, you will see it is completely a fully or predominantly bioeconomic system. And that is the challenge for the synthetic biology too. And so there's a lot of regulations are required, a lot of things are required to go there. So in one way, if you want to define now what the synthetic biology is, so one summary I can tell in that way that, so here if you see this picture, it is nothing but the evolutionary, that's the diagram of the evolution. Okay, it's a toy diagram of the evolution. So this is what the 4 billion years which has happened on the face of the earth, and that is the biology we know today, and that is that what we call what exists, right? And synthetic biology, you can tell in some way that what can exist. Now you can see that what biology is today is a subset of this what biology can exist, right? What can exist. And unfortunately, we do not know what is the boundary. This boundary could be very small. We cannot make much other than the what already evolution has already made for us. Or this could be very broad. We do not know yet. But this is a very important problem to understand that what can exist and what would be its boundary. So this is a fundamental questions we can think about, or the philosophy of the biology at this moment. Now we change a gear a little bit. Now we are coming to a point that if you in your lab, if you really want to make those kind of the synthetic genetic circuits. Okay, how you are going to start, what things you need to study, and what kind of experiments you have to do, right? So here I'm just going to talk about one of our recent publication where we tried to develop a circuit, actually a combinatorial circuit, and the circuits actually look like that because it is a complex circuit, and our goal is to make this kind of the complex circuit in a very minimalistic way in the single cell. And how we are going to do that? So here I'll just explain one thing, that if you see this circuit, we made it, there's a this gate, okay, here, this is an AND gate, okay, a 3-input AND gate. You can see there is a 3 inputs and it can give a 3-input AND gate, and we'll try to see that how you can actually develop a 3-input AND gate in actual molecular terms. So first I'll tell you the results. So this is the whole circuit, and this whole circuit has a 3 inputs, chemical inputs, ATC, IPTG, and AHL. Those are all small molecules, and these circuits are biocircuits actually made inside the bacteria E. coli. And this is your inputs, and here is the output. So if this circuit has this kind of the input and this kind of the output, and we develop the circuit, put in the E. coli cell, and then when do this experiment, you can actually see this is the results. If you cannot understand directly from here to connection, just trust me for the time being that exactly what we are seeing here, we are seeing actually in the experimental. So we actually developed that. Now the question is how we have developed that. Okay. So developing any circuit, you actually need to part of things. So the whole idea of the synthetic biology, as I said, you start from genes and the proteins, and then go to the logic gate level, you make a device, and then those device you connect them, you make a system. Okay, that's the idea. So you start, so when you do a cloning, so I think whoever has the background in the biology, maybe who has the background in the physics, they do not know a lot of things, but just but you probably know something called the cloning. And if a gene expression has to happen, gene expression means if a gene has to produce a protein, and then what you need a promoter. So the promoter is a part of a DNA sequence, okay, just before the gene. That promoter actually, their RNA polymerase actually binds here, okay, and some transcription factor, some other proteins can bind here, and it start producing an mRNA, okay, from the gene, and that mRNA is going to go to the ribosome. It is going to do the protein. So if you want to engineer, we engineer in the DNA code, and in DNA code, the first part is that promoter, and after that promoter, there is something called ribosome binding site. So this is again a part of this DNA. What this ribosome binding site? So when this what this ribosome binding site will do, when you have an mRNA transcribed from here through the RNA polymerase, so this sequence, what is an mRNA, will bind with the ribosome, so that your translation from mRNA to the protein will happen easily. Okay, so what is the so if by changing this ribosome binding site, means that by changing the interaction between the ribosome and this mRNA, you can actually change the translation rate. So that is why it is very important, because you're dealing with the kinetics and a lot of parameters here. Next is the gene itself, which is going to produce the protein. And there, after that, there is something called transcription terminator. That is very important, because if you have a promoter and this RNA polymerase is coming, RNA polymerase can go beyond this. Okay. Now, problem is, if there is some gene is here, okay, another gene in the downstream, if you have a strong promoter, then at the same time, you are not only producing this gene, but you are producing the other gene, which may not be required at particular time. So to stop that, you need a code which is called transcription terminator. That actually terminate the transcription process. So this is in your part, these 4 parts are very important to make a genetic circuits, and you can change a lot of the time their code, and that is the way you are going to do that. Now, when you have the part, you engineer that part in such a way, will come how, then you make a device. So device means you make a logic gate. It could be an AND gate, it could be an OR gate, it could be NOR gate or something. Okay, will come in a moment. And when you have this basic gates, by just changing the sequence in such a way, then those gates will connect together and you make a system function, whatever you are going to be required, right? So this is the idea. So here I'll ask you a question, not to answer that question, but to think about that question. This is again a fundamental philosophical question for the biology. So you think about that. We do not know the answer. So when you actually talk about this kind of engineering, we are thinking that biology is a modular system, like the computers. You connect them and actually you make a system like that. But really, biology is a computer. If you see the evolution, we can see that if you actually compare the genetic network from one organism to another organism, it sometimes it is a very continuous process. There is no modularity. Okay, but when we are engineering this system, we can at least at this practical level, at a certain experimental level, we can look at this in the modular system and connecting them and developing this as a modular. Okay, so sometimes if you don't think about the synthetic biology, the other biologists who are working on the evolution or much fundamental biology, they believe that the gene expression unit itself a modular. Now the question is again philosophically that if either biology is modular or non-modular, or if it is a modular, at what extent it is modular. So, okay, so these are the very fundamental questions of the biology this century is asking for that. That was not probably the last century we asked those kind of the question. This is the century when we started asking these kinds of questions very seriously to understand the biology or to develop the theory of the biology, which is yet not there for many things. Now, here will come. So here is that 3 input AND gate. That is the one of the part of our circuit what we showed, right? And this is the truth table of that. So AND gate, what is said, only when these 3 chemical are present, then this EGFP or the green fluorescence protein will produce. And if you want to see that here in the experimental thing, that is exactly happening like that. So when you have all these 3 chemicals are present here, then you can see there is an increase in the EGFP expression. Any other cases, either it is 0 or it is the very low. So in reality, it is actually giving you a 3 input AND gate circuit. Now I'm coming directly that how you are making it. So in a paper and pencil, you design a completely new promoter. So this is one way we develop. There's a lot of other ways. So if I explain this, you can actually get that what could be the other way you can actually develop this. So here we develop a completely new promoter. So what is this new promoter has? So generally promoter has a 2 hexamer boxes. Here it is, you can see one is the place is -33 and something called -10. So here is the hexamers. This hexamer is called Tata box, where the RNA polymerase binds. Okay, so here is the RNA polymerase binds, and then the TSS, it is called transcription start site, from where the transcription happens. And this transcription, if you have a gene, so it will transcription happens from here. It takes the RBS and the mRNA, and the gene, and it's produce an mRNA. And this mRNA goes to the ribosome, bind with this ribosome binding site sequence, and this translates the EGFP. So now you develop a new promoter. You do not know anything except that you know this Tata box sequence, and now you place something called transcription factor or operating site at different transcription site, means transcriptors operating site in such a way that, for example, here between this -33 and -10, we put a sequence of or the operating site of the LAC-O1. So LAC is a kind of a protein which is called depressor sometimes. That protein or transcription factor, if it binds here, okay, because the DNA sequence it binds here, then RNA polymerase cannot bind due to the static hindrance. That means if the in your system, if you have this LAC-I and it binds here, it cannot produce any gene, right? Similarly, just after transcription start site, we put 2 sequence of something called TET-R operating site sequences, and TET-R is another transcription factor which actually if binds here, then RNA polymerase can bind here. Let's say there is no lac, RNA polymerase can bind here, but RNA polymerase cannot go forward, right? Because in order to transcribe an mRNA, it must go forward. But here it cannot go forward because the TET-R proteins are sitting here. Okay, again, there's a steric hindrance, it cannot go, right? And then at the upstream, there is something called a Lux box. It is another system, and here there's a there's protein from the Luxer family. It binds here, and if you put an chemical, it change the conformation in such a way it helps this particular RNA polymerase to transcribe. If this is not present, the RNA polymerase won't transcribe. So now there are so here I just says that AHL, that is an small chemicals, and this AHL is required in with it needs to bind with this Luxer protein so that the RNA polymerase can properly sit and helps this whole system helps this RNA polymerase to go here. Now there is something molecules called IPTG. These IPTG molecules, so what will happen? This laci, they basically make tetramers, and those tetramers as going to bind this lac one side. And if this IPT, so and it cannot go there. Now, if you add IPTG to the system or to the bacteria, this IPTG will bind with this laci complex. And when bind with this laci complex, it changes into a conformation in such a way that changed conformation cannot bind with the LAC01 operating site anymore. That means LAC01 will go out and your RNA polymerase can sit here. At the same time, there is another chemical, so it's called ATC, and this ATC will bind with the TET-R. So because if the system, in your system, if you have a TET-R, this TET-R will already bind here. RNA polymerase cannot transcribe this EGFP, but if you give the ATC, ATC binds with this TET-R, change their conformation in such a way, now it cannot bind it here anymore. So and then RNA polymerase can transcribe. So which basically means in order to the AGFP to express, you must need all these three chemicals, IPTG, ATC, and AHL. Only then RNA polymerase can actually start transcribing the EGFP. So here, this promoter sequence, it does not exist in nature. So you design either in your paper and pencil or in the computer, and then you synthesize this promoter, you connect with the EGFP in the cloning, do this kind of the experiment, and you may see this kind of the behavior. Now, even let's say when in your first try you develop this design and you get this kind of the behavior, does that mean that this is giving an AND gate? The answer is no. So I'll just go in a writing mode and I try to show you what I'm going to what do I mean by that? Okay. So okay, so I hope you can see this Microsoft Paint. Okay. Now here I am using one something called Wacom, and I just apologize because I cannot write very well in the Wacom because I'm writing in a small board and I have to look at the screen in order to see if my writing is getting clear. Okay, so please excuse me if you cannot, I mean, if you have a little problem in seeing my writing. Okay, so now, so what we have, so what I said that we need three chemicals, right? IPTG, okay, ATC, and AHL. If three of them are present, then you see EGFP is going to get produced. So how you are measuring the EGFP is very simple. You are actually looking at this fluorescence. Okay, that is very simple. But you can change the EGFP with any protein, and that you can connect those device with the other device. I mean other gates, and then the function will go on. Okay. Now there is a, let's say I do one experiment when you have all these things ready, and I'm measuring the EGFP concentration or EGFP fluorescence. Okay, that is in the y-axis. And in the x-axis, I have, let's say I take this chemical, ATC. So, and concentration of ATC in my x-axis. Is the x-axis. So, one possibility is that if I start giving the ATC into the system, okay, in the media, you will see that EGFP is increasing this way, right? This is in a linear increase. But so if your system is behaving like that, definitely you are not making an AND gate. AND gate said, so in generally, so it's a digital logic, right? If it is going from a zero, I mean 0 to 1 or whatever in the linear way, it is not, it is a continuous thing. So it is not that the logic where the 0 to 1, when you actually give the ATC, you see there is a EGFP expression, and if there is no, it's not like that. It is going very this way. What you need behavior, I mean, if you think in terms of logic gate and actual electronics, so here this will behave like something like that. If you see any electrical engineering book or even in the Google, so you'll see. So this is a very sharp switch. This sharp switching is important. Then that is called the digitality, right? That is the digital thing you require. So that means if your system in your biological system, if you give the ATC and your EGP is increasing linearly, that is not giving you an AND gate. Even the previous thing I have shown that giving this kind of the behavior. So what you need this kind of switching behavior. In reality, what biologically you will see, I'll also show you from the real data, you see something like that. Okay, so you never see this kind of thing, but you see this kind of the sigmoid kind of behavior. Okay, so now what kind of, so what parameter is going to tell me that now this is some digital, right? It is not the linear response, but it is some digital. What kind of the parameter? I'm coming into that. And so now if I want to develop that 3-input AND gate, means I have to get, I have to experiment for each of these chemicals, ATC, IPTG, and AHL, and each and every cases I must need this kind of the sharp transition. Otherwise, what I am calling is an 3-input AND gate, it is not an AND gate. Okay, that's important. So how, in terms of molecular engineering, how we are actually developing this kind of the system, right? So, okay, first I'll go is something reference. So you probably heard this function, and this is called Hill function. Okay, so what is Hill function? So if you see here, so real biology, you will see a sharp transition, but not exactly this kind of the sharp, but you will see a slopey things, and it is coming there, right? That is, as I showed it here, right? That is, you can see in a lot of biological system, and sometimes it's called the sigmoid curve. This kind of the sigmoid curve can be explained by a mathematical equation called the Hill function. And if you see, if it's a y and the x, the simplest form of this kind of system, of this equation, would be y equal to x to the power n 1 plus x to the power n. Oops. Okay. So that is the simplest, that is the Hill function. Hill function could be also like, okay, here. So let's say it's another application, forget about this AND gate, but in another application, maybe your protein or metabolite or something, instead of going this kind of transition, it may come this kind of transition. It should come like this. Okay. In that case, your equation or Hill function would look like the in simple form, it will look like 1 by 1 plus x to the power n, where n, either this n or that n, this n, it is called Hill coefficient. Now, in order to get this digital nature, okay, your n value must be greater than 1. If n is equal to 1, how this thing will look like? This thing will look like something like, oops, sorry, please go and it will plateau something like that. So this is, you have seen this enzyme kinetics kind of the things. So it is looks exactly like the enzyme kinetics, where the increase is kind of the linear, then it's actually you make a plateau. Maybe my drawing is not good, but those kind of thing where n is equal to 1 and that's linear, and you cannot take, if n is equal to 1, then you cannot take this is digital. And sometimes there is a term people use instead of digital or degree of digitalness, people use, that is the mostly used in the synthetic biology, and called ultra sensitive. So if n is more than 1, okay, so we call this basically ultra sensitive. So generally I would say, like, I mean, many cases you get n is equal to 1.1. The question is, can you tell is the ultra sensitive or digital? I mean, there's a debate on that, right? But it depends on your functionality or something. But anything near 2 or more than 2, I would say you can actually tell this is definitely digital in our terms. Okay, so that's, so you need this Hill function. Now, question is, which basically means that when you have your circuit ready or whatever construct ready, now you do the experiment, okay, in best of this, you with your chemicals, you're trying to measure your output. It output could be protein, or it could be metabolites, it could be any other biomolecules. If you are going to do that, then what is important, then you actually fit those things, right, with this kind of a Hill function. So what kind of equation, I am coming into that, but the general nature exactly look like that, and fit in the equation and try to find out if your n is more than 1. If it is 1, okay, you are good. So now you have a circuit which is probably ultra sensitive or digital, and you start using it in a different way. Now, so if you see, that's the how this synthetic biology actually start modeling those kind of things. So I'll just go what, when, like 2000, 2001, 2005, 2006, 8, something. It this, like, I mean, that time era, the way synthetic biology tried to model a biological reaction and tried to give their prediction and something, it is different than what we do or try to do it today. Today's is a little simpler, and that's probably required. And just, but I go what it will look like in previous way, and how to write equation for that. If you, so I understand that a lot of you are from a different backgrounds. It doesn't matter if you are from a physics or a completely biology background, but I'm just going to tell you just some small simple thing. Now, okay, now so what is happening? So you have a promoter, right? This is AND gate promoter or whatever promoter. Let's say I name it as a P3, and this is going to produce something called EGFP, that is the enhanced green fluorescence protein. Now, and then a lot of other reactions are going on. So now how we model this kind of, what are the chemical equations we can write for this chemistry, right? It is in one way, it is chemistry, what is happening in the chemical reactions. You can call it biochemical reactions, but nothing but the chemical reaction, right? So now how it is happening? So you can write in this way. So you have a promoter, P3, and now there is an RNA polymerase. Oops, RNA polymerase with binding with this O3, and you give a rate constant K1, and what it is going to produce? It is going to produce mRNA. mRNA for EGFP. Right. Now, just remember that when we're writing the biology, this kind of the reaction, there's one thing. So when in a chemistry, you always write something like A plus B, and it goes to C, right? Or C plus D or something. But here, your promoter is there, RNA polymerase this and mRNA, but this RNA polymerase is not degrading. I mean, here B, A and B is producing C. Probably A and B are connecting the two atoms and making a molecules of C, right? So this A and B are getting consumed. In this reaction, RNA polymerase and the promoter is not getting consumed. So therefore, here you have P3 coming back, right? And RNA polymerase is again coming back. So it is your chemical equation 1. So now what is happening? So what is happening? Now you have an mRNA. mRNA for EGFP. And this is going for with ribosome plus lot of other things, tRNA and so, right? And this is going to give you an EGFP protein. So I just write an EGFP. So this is your equation 2. And what happens inside the cell? This EGFP, this protein, it degrades. Okay. So here is the K2 is the rate constant. Here is the A3 is the rate constant, and it degrades. It's equal to equation 3. Okay. But this is not the only 3 reactions are happening. If you see the other reactions, okay. So other reactions means now you have laci. So how, so, or this laci is the n amount of laci, generally 4 of them. Laci is going to make complex LAC4. Similarly, there is a tatar. So it has a equilibrium constant K1 and K2, where this N tatar is going to make tatar a complex of the tatar. Okay. Similarly, you have this Luxor, they are going to make these things. At the very same time, there is a, okay, I'll go another. At the same time, you have your P3, that is the promoter. With this P3, your laci complex is going to bind, and you get some as a, let's say, P3 S1. Just a name, nothing but, right? So you can give something here. Similarly, this P3, it is going to bind with your other transcription factors. It is going to do something, and so on, so on, so on, right? And then other set of reactions are happening where this laci complex or any tatar complex, this laci complex, it is going to bind with the IPTG. It is going to give you a form, some form, which is not going to bind with that things. So you can see, if you just want to, this gives these 3 things. But again, another thing is that in the cell, we have some promoter from where this laci is producing, tatar is producing, the Luxor is producing. Okay, that I haven't shown in this promoter design because those are just constitutively producing. That is, but the whole circuit actually requires that. So for that laci production, you need another equation. For tatar, you need another equation. Not single equation, but the transcription translation. So you can see this is a series of equation, and each of the equation you can actually convert to a differential equation the same way for a first order kinetics you have done in a in chemistry, right? First order reaction and something. So if you do that, so you have now in your chemistry, when you actually talk about a reaction A to R, okay, and then you just talk about a single differential equation, just a single differential equation. You solve it and you get what something called the first order kinetics. Now here you can see there are several series of reactions. So for the series of reaction, you have series of differential equation. Okay, so you have a series of differential equations to solve simultaneously. And many times that is very difficult in many aspects. First of all, you have to know the rate constant. It is very difficult to know the rate constant for each of them. Okay, so then you assume something and those kind of things, and then definitely you cannot analytically solve them. Definitely you have to solve them numerically. So we used to do this numerical solve using the MATLAB, and many times when you have a series of the reaction, those kind of numerical solve got unstable. But synthetic biology actually went through those painful era. Okay, one important whenever you, so all these things, so there's a, so how we measure that when it's a differential equation, we are not measuring the dynamic. We can measure the dynamics, but what we are doing for a total circuit is something called a steady state. Okay, so you have three reactions. If you just take this first three reactions, that is your mRNA, I mean your EGFP is producing at from some point. Okay, and then EGFP is degrading. Okay, it is degrading. So if you want to develop this equation, so basically your first equation is nothing but dEGFP dt K1 for what EGFP is producing minus something, let's say D1 EGFP. Here is some terms. Okay. And now, so this is the equation, single equation, but this equation has all these simultaneous equations together. So basically in steady state means something is not changing with time, means dEGFP dt equal to 0. So if this is going to be the 0, then your EGFP concentration at the steady state would be equal to whatever the K1 term is here by your D. So this term is going to tell you about this EGFP concentration at the steady state, and exactly what you are measuring this EGFP concentration. Okay, so experimentally what you do, you actually take that particular circuit and you give the chemicals into it, right? And you start measuring the EGFP concentration after some time interval. And what you will see now, so what you will see that this EGFP is going to increase. Okay, and this could be increased in many different ways, and at a some point it is going to kind of plateau. Plateau means that your EGFP is not changing with time. So this is your steady state, right? So this steady state, you are measuring this concentration, right? And put in your mathematical equation to actually, I mean, so that is the connection between the mathematical, your equations and the measurements. So you can measure this, right? Okay. But just remember, this is, I mean, it is not, I mean, this is normalized with the bacterial growth, or you can think of it as a single bacteria, because when the number of bacteria will increase, your EGFP will increase, and that's not the case. It is normalized with the bacterial growth rate, and then you can, it's not just the bacteria, but any other cell, if you use to that, then the steady state. So that is the connection. Now, but what I'm trying to tell you, the point I want to make here, that if you do this kind of modeling, this modeling would be much more difficult, because it's a series of differential equations. Because I remember during my PhD, I have a very small system tried to develop in the E. coli, and in that case, there's a 33 simultaneous equation I had to solve. And but the system I chose is very well studied in vitro. So we have, I had access to a lot of kinetic data, and many of the cases we have to measure those kinetic data and so on. So, but if you think about in terms of the engineering, so if you develop a simple system, you have to go for like a year on the mathematical modeling, getting this kinetic data, then this is not going to be an engineering. Engineering has to be things very, very simple. Okay. And so at this moment, what most of the people will do, definitely that doesn't mean that people don't do this kind of the modeling, they definitely do. But what the, at least our lab and many other labs who want to go a little easier, so how we actually develop, we again, we think about this, what is called is the Hill function. Okay, now that's, I'm going to just switch. Okay. So we do. Oops. So this is our Hill equation. Now you take this equation. Now it is so simple. This is a single equation you can see. So forget about this IPTG and AHL thing. Just concentrate on this ATC thing. So you have dEJP dt, which is equal to some constant, plus concentration of the ATC divided by some capital K with ATC to the power n ATC, which is same as the n in the thing, and then n in the Hill equation, and then 1 plus ATC by K ATC into the n, right? So this is the Hill coefficient. Forget about this. Just think about that if our system have something EJP is producing in terms of the ATC, it looks like that. So what is this constant? So this is called Hill coefficient, as I said, and this capital K, it's called Hill constant. This is Hill coefficient, and this is called Hill constant. And now there is another constant I put it here, and what this says, this is basically a basal level expression. What it means that when you have your promoter, your engineered promoter and EJP ready, when you do not have any chemical, you should not get any EJP, right? But in reality, there is always a leakage. Means some EJP is producing. Okay, you cannot actually, because everything is designed by evolution, and here is a human evolution, which is not definitely not good as the evolutionary design, right? So that the way evolution or nature has to be changed. So there is always, you get very small amount of the EJP's leakage. Okay, sometimes it is not experimental observable, sometimes it is. So you have to just take this into account. So now your equation is nothing but this minus dEJP. Actually, we know that when we are developing one synthetic gene circuit, it should give a behavior like the Hill function. So now if you tell your mathematical or your equation in such a way, so all your reactions, like all reactions has a lot of rate constant and something, everything is now embedded, okay, within either this n Hill coefficient or this Hill constant. Okay, so now many people, so if you, I mean, okay, if it is a systems biology class, I could have shown that you take all those 30 or 33 equations, okay, just by write the differential equation, and you can actually convert them to a similar kind of equation. So all the rate constant and everything and now go there. I do not know that what is the constant here. That's why we do the experiment. We feed this equation and we actually get those things. So now this is a differential equation for 3 of them, and U minus d EJP. D means that degradation of the EJP. So when these twos are same, that is means when dJP is equal to dt will only happen when the production of the EJP and the degradation of EJP will be the same, and you actually equate with the 0, and what you get at the EJP at the steady state, it looks like this equation. Okay, now it is much simpler, a single equation. It doesn't have this 33 or something equation. And now when you have this kind of equation, you basically fit those equations. You fit those equations experimentally, because you have the equations, and when you fit, you get all the constants, right? For here, for this particular situation with the AHL, we got the Hill coefficient is 2.97. For with ATC, we got 4.65, and IPTG, whatever we got. So this is the 3 you can see, and all the 3 cases, this coefficient is greater than 1, which says for all these 3 cases, this is ultra sensitive or in the digital in nature. So when you get this, so okay, it's good, very good. So now we want to do a simulation that how it would look, and we have to retest or validate the simulation to see that what we can predict something for our gene circuit, and the similar behavior is coming. So here is just the real data. So now when you have from these experiments, you feed those equations and you get that exact parameters. You put those parameters back to the equations, right? The simple Hill equation, and then you run a simulation in the computer. And when you run the simulation, what you get this kind of the behavior. So if, I mean, HL and ATC, you can see that, and when your IPTG is constant, here when your AHL constant, how the behavior looks then in simulation. And then when you got the simulation, we do a retest, or to validate, we actually do an experiment. Now, if you see this simulation and the experiment, all these bottom panels are experiments and top panels are the simulation, you can see there is a very good topological match, which basically now telling you that whatever the gene circuit you actually developed, it is predicted, because you predicted by the simulation, and now you can validate BIMS. It is, I mean, predictive simulation and the experimental, it will match. So this is a predicted model of a simple circuit. So this is a 3 input AND gate, and then the actual paper I showed, there is a other gates are connected, and then we forgot. That's I'm not going, but this is the general way. But one thing I forgot to tell you, that it is not just when you made this your circuit and then you get this on all those results. So when you made this circuit, you will get a lots of leakage. Most of the cases you won't get the proper N values. So you have to optimize those circuits. Okay. So in today's class, we are not going to talk about this optimization. In the next class, I'm going to talk about the optimization. But one important, so if you see the way we are doing, so you have a design. So you have the design of the 3 input AND gate or the whole circuit, and then we start building it. We build this 3 input AND gate, and you test it, right? And most of the cases, when you have to test it of the first time, it doesn't work similar to as your design. Then we learn from this something. And when you learn, we go back to the design. You change the design in such a way. Again, we build and again you test. So this is called a design, build, test, learn cycle. This is the cycle for the engineering. So any engine, like when you make a car, when you make some plane, so you always come through this kind of the things. You design, you build, you test, you learn, you design. So this is the cycle of, it's called the engineering process pipeline or engineering cycle of optimization this way. So how we are going to optimize that, and how from those kind of gene regulation, what is the connection between this Hill coefficient and the molecularity, why it is developing those kind of nonlinear function. I think I'm going to talk about this in my next class. So for now, I think that's all for today. But definitely, if you have any question, please go on, and I'm happy to answer those questions. So if you do not have any question, then I think we can stop here.
So no question.
Okay. So then I think we stop here. Thank you very much for attending.
Thank you, sir.